floor is yours.
I don't know. Have you contacted Bu Nunun before, Hannah? If maybe she found some problems. I don't know if have you contacted her before?
Sorry, so maybe she is currently in the field, so she has a problem in connection.
Yeah, okay. Yes, the greetings come from me. It's okay. Yeah, hello. So good afternoon, everybody. Good afternoon, Rafael Perez. Thanks for your coming here online. And today, students, you will have some lectures from Dr. Rafael Perez. The moderator will explain a little bit the CV of Rafael Perez, I think, but the point of view from my side is not as lecturer in modeling, but as the Vice Dean one. I don't know if your explanation will be deep or not, but I think it's okay. The discussion it will be started by student. I think, yeah. Okay, I think that's all for the moment because the time is so limited for this meeting, and I give back to Hannah. Thanks, Hannah.
Yeah, thank you, Mr. Afif, as our Vice Dean 1 for representing our department and for the engaging greetings. We are privileged to have Dr. Rafael Perez, a renowned expert in this field, joining us today from Sirat, France. Before we begin, let me go over the CV of our speaker. I will introduce our esteemed speaker, which is Dr. Rafael Perez is a distinguished researcher and holds a PhD in agronomy and crop science from Montpellier Sopagro. He holds a master's degree in applied science degree in agronomy in ENSAT. He also specializes in combining modeling, plant breeding, and ecophysiology to assess the development of crops' environmental interaction. His recent research was in 2018 of postdoctoral research about analyzing and modeling the genetic variability of corn architecture in response to water, stress, and competition for light in French National Institute for Agricultural Research. And surprisingly, he is also able to speak Indonesian. With his extensive experience and expertise for plant modeling and ecophysiology, Dr. Perez has made significant contributions to the field of sustainable agriculture. We are honored to have him here today to share his insights. Welcome and good afternoon, Dr. Perez.
Good afternoon, selamat sore. Thank you for the invitation. So I can start sharing my talk. Is it okay for you?
Okay, Doctor, because that was a brief introduction, and
Yep.
the virtual floor is yours. Please proceed with your presentation.
Okay. Okay. Let me share the screen. Okay. Can you tell me if it's okay now?
Not yet.
Not yet?
I don't know
Okay,
if
sorry,
you are possible to open your slides by your Zoom.
Yes,
Okay.
I'm doing it, but I think I have a security on my computer, and maybe I need to reconnect. So you can't see the presentation now?
No, there is
Not
no
yet.
slides.
Okay, I'll try again.
But if it doesn't work, it still doesn't work, you can send me Rafael and I will send to the host of the meeting to be shown in the Zoom. But up to you.
Sorry, I had to start again, I think. Can you hear me?
Yeah, okay.
Is it okay now?
Yeah, yes. Okay, very good.
Okay. I will just put, okay. So can you see the red dot? Because probably I will use it for the presentation.
Yes, yes. The lesser
Okay.
point. Yeah, the lesser point.
Okay, so thank you for the invitation. So I will talk to you about the work I'm doing here in CIRAD in France. In modeling, in plant modeling and ecophysiology, and I will present you different topics of research I am conducting and to to show you how we can use plant modeling for different purposes. So don't hesitate to interrupt me if something is not clear. Okay. So the title of this talk is Using Plant Modeling as a Research Tool, and I will focus with example on functional structural model of oil palm. So to start, as you all know, plants needs resources to grow. Among these resources, you have the carbon dioxide, sunlight, water, and minerals. Okay. In this talk, I will focus on the sunlight. So how can we improve plant productivity? Plants grow in a given environment. So usually we cannot change climate variables, except when we have some management practice where you can add input in a resource like water or fertilization. You can have cultural practices as well that change the environment, and the other the other way of acting on plant productivity is using plant breeding. And what is interesting is the interaction between these three factors. So regarding the light resource, the way to improve the plant performance acting on light interception is either to select specific genotypes that have some capacity to intercept light, or we can act on management practices, mainly with cultural practices like pruning or planting design and so on. So if I come back on some researcher, I can introduce you the concept of ideotype. So an ideotype is a plant where we want to select the best behavior in a given environment. So for instance, if you look at the selection of rice, the traditional plant type was like this, with a given architecture, with lots of leaf area, tall plants. And within the years of selection, we reach a new plant ideotype in China, for instance, that is called a super hybrid rice, which is smaller with erected leaves, and that is very is used and at very high density. So there is interaction between the planting density and the morphology of the plant to improve plant performance in relation with light interception. So how can plant models help to improve light interception efficiency? So first, how we model yield in crop yield. So the basic formalism is the Monti's formalism, where you have the light environment here, so the photosynthetical active radiation, and so the yield is the sum of all the light that is intercepted during the plant cycle. This light, this amount of incident light, is intercepted with a given efficiency, which is called the light interception efficiency. This amount of light will be used to produce carbon with an efficiency called biological efficiency or carbon assimilation efficiency. And once the carbon is assimilated, it's partitioned within the plant between the vegetative part and the reproductive part. with which is called the harvest index. So the yield is the the product of all these factors. And so today I will focus on light interception efficiency and carbon assimilation efficiency. So there is two types of crop models. First, you have what we call the process-based model or crop model, where then there is no representation of the plant architecture. In crop models, we have what we call the leaf area index, which is the amount of leaf per ground area. So there is not an explicit representation of the plant, but the canopy is represented as a medium. It's an amount of leaf area that intercept light, and all the physiological process are modeled with this assumption that the canopy is a homogeneous medium. The other kind of model is the functional structural plant model, where you have an explicit representation of plant architecture in 3D. So we have to model the architecture of the plant using architectural model. So in both model, we have to model the light interception. In crop model, we use what we call the Beer-Lambert law, which comes from the optics. And we estimate the capacity of the canopy to intercept light with a given efficiency that depends on the leaf area, so the amount of leaf area you have in the canopy, and the other factor like the clumping index, or I will not go into detail, but it will helps to model the capacity of the canopy to intercept light. Okay, and in this model, the output is the fraction of light intercepted. So you have the incident light coming from the sun, and here you have this incident light is intercepted with a certain efficiency, and usually we use the fraction of light intercepted. So this amount of light intercepted is estimated at the plot scale, since we have not explicit representation of one individual, but the whole canopy. In FSPM, Functional Structural Plant Model, we couple the architectural model with a light model, where we have an explicit modeling of light, where we will cast some rays, sunbeam, on the 3D plant. So in this kind of model, we have a fine resolution of the quantity of light that reach each part of the plant. So we have resolution at the organ scale, but we can upscale the results at the plant scale or the plot scale. And so in both models, we have what we call the light interception efficiency, that can be either the fraction of light intercepted in crop model, or that can be at different levels in FSPM. Then, when we have the quantity of light intercepted by the plant, we can convert this light interception into carbon using a photosynthesis model. In crop model, we'll use radiation use efficiency. So the radiation use efficiency depends on the plant, on the species, and it's the relation between the total dry matter produced by the plant and the total intercepted solar radiation. So here you have some example on different species, and the biological efficiency is the slope between these two variables. So for instance, you can see that apples, for a given intercepted solar radiation, will give you a total dry matter. Barley has a higher efficient biology, and so on. So this linear relationship is true when we integrate it at the crop scale over a long period. But in FSPM model, there is not this kind of assumption, as we have the explicit representation of the plant and the amount of light intercepted by each organ. We can estimate the amount, the production of carbon assimilated at for each leaf, because each leaf will have a different amount of light intercepted. For instance, if you are in the upper part of the plant, you will be directly exposed to the sunlight. But if you are at the bottom of the plant, you will have self-shading of the plant that will reduce the quantity of light intercepted at the leaf level. So in each leaves, we can estimate the amount of carbon assimilated using a Farquhar model. So it's a photosynthesis model, and it takes into account the relationship between carbon assimilation and PFD, which is photosynthetical photon flux density. Okay, so in each leaves, you will have a certain amount of light intercepted, and we will convert it into carbon. And we can then integrate all the carbon produced at each leaf at the plot scale, at the plant scale, and then at the plot scale. So the main difference in the two models is that the crop model can only model physiological processes at the crop scale, and in FSP model we can model it at a finer scale from organ to plant or plot scale. Okay, so now I will give you some example of FSP model on oil palm, and with different examples. So first, I will show you how we can use FSP model to assist plant phenotyping on complex traits. So this is the work I have done during my PhD, and the question was, how does oil palm architecture vary, and how does this variability impact on light interception? So for instance, we were working in we had different progenies or genotype in the field with variation in their architecture, and we wanted to know how the difference in architecture impacted the capacity to intercept light and then to assimilate carbon, and then plant performance. So the first thing was to evaluate how architecture was different between the progenies using field measurement. Then we had to model the plant in 3 dimensions to simulate light and to estimate the efficiency to intercept light according to the architecture of each genotypes. Okay, so this is the pipeline where we have field measurement, we reconstruct in 3 dimensions the plant, and we simulate light interception on this 3D mockup. So first, how we can evaluate architectural difference among progenies. So to come back on oil palm architecture, so oil palm is a corner model. So there is one shot, it's a monoaxial shot with indeterminate growth, and the inflorescence are lateral and can be either male or female. So the production, the oil production, come from the female inflorescence, what we call a bunch, and we extract the oil from all the fruits from this bunch. Usually, the conventional planting design is 136 plants per hectare with a King Kong's design like this. Okay. So when we arrived in the field, we had to measure all the architectural traits, which are mainly leaf traits in oil palm, because there is no branching. So if we looked at what have been done in the literature in terms of genetic control of plant architecture, lots of studies showed the inheritance of plant 8. Okay, so you can control plant 8 with genetic. There is difference between genotypes. The branching pattern can be modulated by genetic pattern as well. And leaf geometry depend on genetics. So here you have some example on tomato. different genotypes of tomato have different morphology of the leaves, and this will impact the capacity to intercept light. Here, for instance, you have different genotypes of rice, and you can see that you have dwarf genotypes, or you can have a difference in in the tillers and in the erectness of the tillers. In oil palm, some studies show that stem height is genetically controlled, the leaf area as well, and the morphology of the leaves. So when we are in the field, we had to measure all the architectural traits, which at the plant scale are resumed at the stem height, the stem diameter, and the phyllotaxy, which is the angle of the leaves, of the successive leaves. At the leaf scale, we measure the length of the leaf, of the petiole, of the rachis, and also the orientation of the leaf. And at the leaflet scale, we had morphological attributes like the length, the width, but also geometric attributes like the angle of the leaflets. So we gather all these data, and we add all these data set for different progenies of oil palm, and we wanted to know which of the, if there was difference in some of these traits between genetic material, and what was the impact of this architectural trait on light interception. So first, we look at the different traits and the difference between progenies according to these traits. What we found was that the phyllotaxy and the stem height was not different between the progenies. But if you look at all the leaf traits, they were all different between the progenies. So here, for instance, you have the ratio between the petiole and the rachis. So the rachis is the part of the leaf where you have the leaflets, and the petiole is the base of the leaf. And what is interesting is that depending on the genetic material, so here is 5 different progenies, you can see that the ratio is significantly different between the progenies. If now we look at the leaflet length, according to the position of the leaf, you will have a given shape. So you will have small leaves at the beginning of the leaves, the leaflets at the beginning of the leaf. And in the middle of the leaf, you will have the biggest leaflet. And when you go until the tip of the leaf, leaflets are smaller. And if you look at the shape of the leaflet length, depending on their position of the rachis, it's quite different between the progenies. So we look at all these parameters, and we keep the one that were different between the progenies, but because we think that if they were different between the progenies, that the assumption that there is a genetic control of this trait. So here is the representation of the 3D mockup we have done for the 5 studied progenies. And the next step was to simulate the development of the progenies within time. So here you have an example of one plant over time, the development, and we can simulate light on this plant. So to simulate light, we use a light model that is called Archimede, that was developed in France, in Montpellier, in CIRAD. And we use the incident radiation. So with a meteo station, we have the quantity of light, of its incident light. And we use this data and the light model to simulate light on how different progenies. So here on the right, you can see the plots of the 5 plots of the different progenies, and the color indicate the amount of light intercepted. So here you have the dynamic of light interception. This is the the fraction of light intercepted at the plant scale. And here is the time, and you can see that depending on the progeny, you have more or less light intercepted. So first, we could assess within the modeling the difference in light interception according to the progenies. If we look at the given moment, we can do statistical analysis and see that we have some material, for instance, DA for Delhi Avros, had a higher fraction of intercepted light. As you can see here, there is more light intercepted by the plant and less light going to the ground. The color indicates you the difference in light interception. So we can prove that there was significant difference in light interception between the progenies. Here is the same result, but it's the light intercepted per unit of leaf area. What we can, the result also was that 20% of the variability in light interception efficiency was not explained by difference in leaf area index. So that means that it's not only the quantity of leaf area that is different between progeny that explain light interception, but it's also the geometry of leaf. Okay, so how the plants position or put the leaf, this leaf area in the space. So then the second question was, okay, we have seen that we had different genetic material with different architecture, with different efficiency to intercept light. Then the question was, okay, but which trait is interesting to study, or which trait explain the most the light interception efficiency? The aim of this was to define ideotype for plant breeding. What is interesting to select in terms of architecture. So to do this, we perform what we call the sensitivity analysis. So we have our architectural model. So the input of the model are the architectural traits, are parameters that give you the quantity of leaf area and how this leaf area is organized in the space. Okay, so you have an explicit representation of the plant in the space. Then we have the light model. So we'll simulate light on this mockup, and the output variable are the quantity of light intercepted by the plant. And the sensitivity analysis is the relation between the input parameter and the output. So we'll see how the change in input parameter will impact output variables. So here the output was light interception efficiency and carbon assimilation at the plant scale. Okay. So what we have done, we changed lots of input parameters. So here is the list of the parameter that, for instance, change at the crown scale. So it's the number of leaf, the phyllotaxy at the leaf scale would be the length of the leaves, the number of leaflets, and so on. And at the leaflet scale is the shape of the leaflets, if they are wider, if they are longer, and etc. So here are some examples of the different architecture we can model with the architectural model over time. So here is the combination of the parameter. And so you can see that we represent some palm trees that are not realistic at all, but it is not the problem. What we want to do, what we want to know is which trait impact the most our output, which is light interception and carbon assimilation. So we make lots of mockup, 880 different architecture. We simulate light on this, and we investigate which architecture maximize light interception and carbon assimilation. So we perform what we call the meta modeling approach. I will not go into detail, but the main objective is to understand which parameter explain the most the variation in the output. So this model helps us to identify the 9 architectural traits that were the most important. So it was the number of leaf on the crown, the length of the rachis, the ratio between the petiole and the and the petiole and the rachis, sorry, and the number of leaflets and so on, and the shape of the leaflets. Here is the final result that gives you for each parameter the sensitivity analysis, the effect of each parameter on light interception, and here it was carbon assimilation. So what we can see is the length of the leaves explain the most the variation in carbon assimilation. Okay, then is the shape of the leaflet, and here in red is the principal effect. It means that is directly the effect of leaf length on carbon assimilation, and here is the interaction. It means that the leaf length alone not explain this variability, but it's the interaction between leaf length and other parameter, for instance, leaflet shape. Okay. So the conclusion that was that erect leaf, short rachis, and petiole, and high number of leaflet on the rachis were the trait that explain the most light interception and carbon assimilation. So using this, we looked at all the mockups that optimize light interception and carbon assimilation. So here, for instance, is the representation of the carbon assimilated at the plant scale according to leaf area index. So here is all the points, so all the 3D mockup with the carbon assimilated by each plant. And what we have done, we selected the plant, the mockup, with the maximum assimilation, and all the plants at a given leaf area index with the minimum assimilation. And here is the the quantity of light intercepted per leaf for all the best and the worst mockup. And you can see that the best mockup, so the max, they don't intercept more light per leaf, but they assimilate higher quantity of carbon for almost all the ranks within the crown compared to the plant that have the less carbon assimilation. Which is interesting because it shows that to maximize carbon assimilation, it's not the plant that maximize light interception that will produce the more carbon, because there is a saturating effect of light. It means that if you have enough light, if you put more light, it will not be beneficial to the plants because there is the a plateau, saturation in photosynthesis. And then we looked at the best plant, the ideotype. So we found 4 types of ideotype with different architecture, but as you can see, they look like a real oil palm. So we can first say that, okay, the modeling approach tells us that you see all the different type of architecture we have modeled. The best architecture in terms of carbon assimilation are quite close to what we can find in the field. So naturally, the architecture of the palm tree is optimized within the design we use to plant it. But we can see that we can slightly change some parameters to get a very good interception and assimilation. So here you have the difference. For instance, you have this ideotype that have big leaves and small leaflet and a erected behavior of the crown. And you have other type of plants, for instance, this one that is less erected, but have long leaflets and small leaves. So there is different combination of architecture that can optimize light interception and carbon assimilation. Then I will show you how we can investigate the effect of planting pattern on system performance. So on, we only looked at the genetic part. It means that how the genetic can influence phenotype and mainly architectural traits, such as leaf traits. But we can also, as I told you, change plant performance acting on management practices. And in Alpine, the management practices is mainly the planting density. So we had another experiment where we had different design. So 5 different designs, different density of plantation, but also different pattern. It means that you can have a King Kong's design. It means that all the plants have the same distance each other. Or you can have design with anisotropic plant pattern, where you have a smaller distance within the row than between the rows. Okay. So here are 2 examples of the density, and what we can do is reproduce in silico this design and simulate light interception according to the design. So what we wanted to know first was, okay, a given material maybe will react differently according to the design. So first we measure the architecture of the plant and we assess if the higher density will change, for instance, the length of the leaves and so on. So what we have done was, okay, I have a given architecture in a given design. So it's the mock-up, for instance, of the design B. We'll put this mock-up in the design B. We look at the light interception and the carbon assimilation in this given design. But what we can do as well is measure a plant and reproduce a mock-up from another design, for instance, the design A, put this architecture in another design and see how it impacts light interception and carbon assimilation. And by comparing the two, it gives us the effect of plasticity on plant functioning. It means why the plant change architecture in a given design, and what are the consequences in light interception, carbon assimilation. And this given mockup, so the conventional in the conventional design, we can as well simulate the mockup of design A in the planting design A. And here we have the difference in terms the effect of the design in plant functioning. Sorry. So first, here I will not go into detail, but what we found that according to the design, the plant were plastic in terms of rachis length. So so here is the neighbor index. It means the distance between trees, which depend on the design. So here each color represent a different design, and you can see that according to the design, the plant have more or less big, longer leaves. Here is the declination of the leaves, and here is as well is how we model the declination of the crowns. It means that if you have an erected crown like this, or if you have a crown open. Okay, so the density change both the morphology of the plant, so the length of the leaves, but also the crown architecture, so the angles of the leaves. So the plant proximity increase leaf dimension, leaf weight, and leaf erectness. Okay, and this was included in our model. So we include the plasticity in the model. And the frequency of the leaflet along the rachis or biomechanical properties of leaf remain unchanged. So we have seen that some traits remain unchanged. There was no response of the plant to the density for these traits. And at the end, we were able to represent each mock-up, so each architecture of plant in each design, and estimate light interception, carbon assimilation. So now, now we had simulated all the 3D mockup and all the light interception and carbon assimilation. We compare the design. So here you have the 5 design we studied, and here in blue is the model that accounts for plasticity, that change. So it accounts for the change in architecture, and in red is when there is no plasticity. That means that we take the mock-up of the conventional design. It means that it's theoretically the architecture of the plant if the plant was no plastic to the design. And so by comparing the design, so the blue bars, we can compare the effect of design on light interception and carbon assimilation. And comparing within a design, the two colors give us the impact of plant plasticity, or it allowed us to understand how the architectural plasticity impact on light interception and carbon assimilation. And we can do the same. So this is at the plant scale, but we can do it at the stand scale. And the conclusion of our study was that the architecture plasticity improved light interception. So the plane changed their architecture to maximize the quantity of light intercepted, and mainly with expanding the leaf area. But at the stand scale, the competition for light imposed by the design counterbalance these benefits. It means actually that each plant changed the architecture, creating more leaf area or bigger leaves. But as the density is higher, if all the plants are doing more leaf area, there is more shading at the plot scale. And so there is no benefits at the plant scale to improve leaf area at the plot scale, at the plant scale. And, okay, so this study was really interesting to better understand what is the impact of architectural plasticity on plant functioning, and what was the impact of management practice, mainly planting design, on plant performance. The other the other thing that we can do is to define innovative design with our model. So for instance, we have a new project where we want to see if, for instance, we were planting palm trees in rows with a high distance between rows that will enable to plant other crops, for instance, rice, during the rainy season. So the idea was to have a kind of agroforestry system where during the rainy season, we will have palm trees and rice within palm trees. And during the dry season, we will have only palm trees, but at a lower density to avoid competition for water. So what we have done, we simulate a lot of design, row design, planting like this. We simulated light, and we investigate the quantity of light that will be available between the rows of light. So in the area where we can plant other crops, and we look at the relationship between the palm density and the quantity of light available for rice, for instance. So here you see that if the plant density is very low, you have more light available for another crop. And what we wanted to to have is a trade-off between palm density, because we wanted to maintain a high density to have a good production of oil at the land scale, but enough light to grow rice, for instance. So we found this design, which was quite interesting. This design is a distance of 7 meters between palm trees within a row, and rows distant by 12 meters. And here you have the kinetic of all the design during one day. And the one that we kept was this one. So it means that you have more than 30% of the incident light that is available for the rice. And then we have studied in control condition the impact of this kind of light pattern on the growth of rice. So now we are with with Professor Afifuddin, we have a new project to try selecting rice tolerant to shade to design this kind of pattern of or agroforestry system. And last but not least, tool that we can use using a model is to understand and simulate plant response to climate. To do so, we perform an experiment in a very controlled condition here in Montpellier in what we call the ecotron. We put some plants, young oil pine, in controlled condition where we can modulate light, temperature, humidity. So, and what we have done, we put the plant in the chamber and we run different conditions in the chamber. For instance, we had different scenarios, so 8 different scenarios. For instance, hot scenarios. That means that the temperature in the chamber will be very high. We have cold scenario where the temperature will be very low. We can modulate the relative humidity. So we will have a dry hot or a wet and hot, and so on, different combination of climate condition and the plant. And what we wanted to do was to evaluate if our model was able to simulate the behavior of our plant. So in this chamber, we were able to measure daily flux of carbon dioxide and water. So here, for instance, I show you for a given day the carbon assimilation of the plant in the 8 different scenarios. So when we change the temperature, here you can see that for very high temperature, the plant have a very low carbon assimilation. In the cloudy, so when there is a few light, so that correspond to a very cloudy weather, we have a low carbon assimilation as well. And here we also in green, we also change the quantity of carbon in the air to see the impact of carbon in the air on plant performance. So the idea was to able to represent this difference in terms of carbon assimilation according to climate. To do so, we had to reconstruct our plant in 3 dimensions. So we use what we called a LiDAR, which is a 3D scanner. We also make measurements with a specific tool to estimate the efficiency, the biological efficiency of the plant. So it's the response curve to light or to carbon for each leaf. It means that when we change the light at the leaf scale, or when we change the carbon dioxide in the leaf scale, how the leaf photosynthesize. Okay. So we calibrate our model using specific 3D mockup with specific calibration of the photosynthesis processes. And we simulate, we reconstruct the plant and we simulate the exact condition that we had in our chamber. That means we simulate this pattern in terms of light, this pattern in terms of water content in the atmosphere, or the temperature. So we were able to resimulate exactly what we have observed in the chamber, and we looked at how our simulation were close or not to the observation. So here, for instance, you have the daily dynamic of carbon assimilation for the same plant in 3 different scenarios. So this is the control scenario. So the best condition for oil palm, 400 ppm. We have a cloudy weather, or we have an improved in the carbon dioxide. And here you can see that the simulation are the red dots, and the observation are the the lines here. So you can see that for these three scenario, we were able to correctly represent the dynamics of carbon assimilation in the plant. But this is some example, because all the work is not yet done, and we have to look at all the other scenario. So this is important, because if we have difference in between our simulation and between our simulation and what we observed, it means that there is some process that are not well accounted in our model. And when there is difference or default in the modeling, that means that there is something that we don't understand and that we didn't represent exactly the good process. So this is very interesting to have hint or to focus on something that we don't understand. So this is one interest of the modeling approach as well, is to know where we have to improve our knowledge. And finally, this is the framework of all the job that is done in modeling oil palm. So we are working with a private company in Indonesia, Smart. I think you may know it, Sinarmas. And so the idea of the project is to have a complete FSPM model of oil palm that will help us to predict the production according to the climate, but also according to the genetic material and to the management practices. So the model accounts for the change in architecture and biological process over time and over space. And we can account at different time in the cycle of the production. We can account for the effect of climate, genotype, and then management practice. And this will help us to design innovative practice, or to select genetic material better adapt to a given environment, for instance, for drought tolerance, and so on. And that's it. I hope it was clear enough for you, and I hope you have some question. Thank you.
Thank you, Dr. Perez, for the enlightening presentation. We will now move on to the question and answer session. As a reminder, please submit your questions using the Q&A feature by raising hands or submitting through the comment section, and we will address them in the order they are received. To other audience that might want to ask a question to Dr. Perez, you can go through the comment section or raising your hand.
So I guess I was really clear if there is no question, or not clear at all.
You can start to give some questions to Dr. Rafael. Hello, Hannah.
Yes, hello, sir.
Yes, maybe you can start to give some question as moderator. You can give example of questions from you. Then
And so far, I have not really quite advanced question, because for me this is an advanced material as well, because I'm still an undergraduate student. But there is something that is interests me. I would like to know like how to actually capture the light. Maybe if there are there any technology that is significant or something like that, because I would like to know.
You mean if there is specific tool to estimate light interception?
Yes, doctor.
Yes, so you can do it by modeling like this. And we at the crop scale, there is some tools, so sensors, for instance, that give you an equivalent of what I show you at the beginning, that give you the fraction of light intercepted. So what you can do, for instance, if you have a rice field, is to measure the incident light above the canopy and measure the transmitted light below the canopy. And it will give you an estimation of the fraction of light intercepted. The problem is by doing this, you have one value at a given position in time and in space as well. So it's why we do modeling, because it allowed us to integrate interception efficiency during the whole cycle and at different and according to the space as well. So this is the problem. The resolution in terms of time and space is quite difficult to harness with direct measurement in the field. So it's why we use modeling approaches.
I would like to have another question. Thank you for the explanation before. Oh, there's a question, doctor, from Ade Fajar Wicaksono. You may proceed your question.
Thank you very much for the opportunity. It's very nice and very wonderful about the modeling for the structural modeling. But I still confuses how to to operate this modeling. And if from I know that I still confused to use this model and to predict the functional of some plants. So can you tell me for the simple way to operate it? And from your presentation last time, that you say that to run this model, we must input some data, but I still don't understand how to transform the data into a structural model. That is my question, Dr. Rafa.
Okay. Yeah. So unfortunately, it's not simple to handle this kind of model because it's a research tool. So it's Afif.
Sorry.
Yeah, so it's quite complex to use it. It's a research tool, so it's dedicated to do research. It's not yet spread for simple use, I would say. So we have different model, and so the data, there is already some script or interface that helps you to input the data and to convert it into a 3D model. For instance, when you have a data file with all the architectural traits, there is some script in R or in different language that helps you to convert it into the architectural model. But yes, when we use this kind of model, it's for a specific purpose, for a research purpose usually. So it's only done by student in either in master with a master degree or PhD students that have specific question and hypothesis, and they want to test it using modeling approach. But so far, it's not. So you need to train a lot to use this kind of model. Yes, so it's not. And so far, it is a cooperation between CIRAD and Sinarmas. So the model so far is not available for everyone, but one day it will be available. Yeah, but to be in the future, I hope that we will be able to give some specific software to run some simulation without coding inside. Okay, I hope I answer your question.
Thank you very much, Dr. Rafael.
Sama-sama. Okay.
There was a question from our audience. Thank you, Dr. Perez. Is there any audience that would like to ask a question to our speaker? You may raise your hand or comment to the comment section and I will address your question. Any other audience, you are welcome to ask a question. Maybe before there's another audience that would like to ask, I would like to ask again to our speaker, Dr. Perez, regarding the previous question that I asked. Actually, I'm a bit curious, is it through this modeling stuff, is there any like a mistake factor that we should take mind of?
What kind of factor you say?
Maybe like the calculation or something like that, that is maybe not quite accurate to the result. I'm just curious on that part.
Yeah, yes, it's true that in all models are false, you know. So in each modeling approach, you have errors, and the more complex is the model, the more errors you can generate. So it's why at the end of the last the last slide, I show you that we are doing a specific experiment to evaluate the capacity of our model to predict some behavior. And if we have difference between observation and the model, that means that there is some errors in the model. So what is interesting and what is necessary is to know the errors of our model, to be sure that when we have some conclusion, there is no mistake on it, or at least that we can know or be sure that when we do, we say, okay, this kind of planting design would be the best. It's the error is lower than the difference between, for instance, the performance on the design. If you can estimate the errors, you can be sure that, okay, I know that this design is better than this one, and within the error that I know, it's true that you should have higher production in this design in reality. So it's important to evaluate the model, to be sure that to have in mind what is the degree of error we are doing.
Thank you, Doctor, for the explanation. We have another question from Ardian Chandra. He asks about, can we use another crops instead of palm tree for your model? You may answer the question, Doctor.
Yeah, of course. Here I present only what I'm doing in my own research, but actually there is lots of FSPM model or crop models in all the main production crops. So on rice you have some, and everything, and wet, and apple trees, mango. And in our team here in Ciarán, we are working working on rice, on mango, palm trees, sorghum, and so we have lots of it. Almost all the crops are already modeled, but I only present on what I'm working.
Thank you, Doctor, for the explanation. To the audience that asked the question, is it enough or is there any feedback to Dr. Perez?
Rafael, maybe you
Yes.
can explain more in detail a little bit for ecotron, because the term of ecotron is not too familiar for students in Indonesia, yeah, especially for our students. Yeah, you know,
Okay.
normally in France, we use some specific rooms that, yeah, like phytotron, ecotron, yeah, this is normal for us in France. But for Indonesian peoples, something like that, maybe we can use also a term that we call, for example, breeding speed. You know, in the room for speed for precision breeding, yeah, for having the progressive of breeding before acclimatizations, we can plan some plan inside of that kind of room, Rafael. But we
Yeah.
don't use this kind of term. So
Yeah,
let's
but
explain a little bit. Yeah.
yeah, actually, Ecotron is only the name of the place where we are doing the experiment. They are all growth chamber. The
Okay.
specificity of these chambers are there is high control on the climate condition, and there is the capacity to measure the fluxes, so carbon and water fluxes. This is the specificity of the Ecotron, that you can control very well all the conditions and you can measure fluxes. But it's growth chamber.
Yeah. So the
That's it.
principle is growth chamber, right?
Yeah.
And then throne. This is specific or the choice of French. Friend, Tom, you can
No, the
explain.
name, I don't know why they called it like this, but yes, there is several Ecotron, but let's say
Yeah.
they are very controlled growth chambers.
Yeah. So yeah. Okay. So Hannah,
Phytotron is
I
the same. It's the growth chamber for plants, but we call
yeah,
it
yeah,
phyto.
yeah. Phytotron, ecotron. Yeah.
Ecotron is not only for plants.
Okay.
They can put everything inside, insects, for instance, and so on.
Hannah, I think there is new questions in the chats, in the meeting chat.
Yes, there is another question from Afranisa Dwi Putri. She asked, what if we use another crops will change the system from your model?
So I'm not sure to understand the question, but the model is specific to except the light model and the photosynthetic model. The architectural model is specific to palm tree. So if you use another crop, you will need another architectural model, but you can use the same light model and Farquhar model, photosynthesis model. The only thing is you need to recalibrate the photosynthesis model given the species you are modeling. Because for instance, photosynthesis of palm tree is very high compared to all the other C3 plants.
Thank you, Doctor, for your explanation. To Afranisa, is it enough? Maybe it's enough for her. To the audience, don't forget to fill in the attendance list below that is already been sent on the comment section.
Yeah.
Is there any
So
audience?
if no more questions, maybe Rafael can give some. That's enough. Okay, thank you for your explanation. Yeah, maybe some concluding remark for the presentation, because the student, I think this, this is the last schedule of they schedule in in our faculty, Rafael. Maybe, yeah, they feel tired little bit.
Oh yeah. So have a good rest, what I can say.
Yeah, but it's okay. Yeah, it's okay for the moment. Several
Yeah, no, but
questions.
it can be
And
hard to follow. I know that it can be hard to follow
yeah,
at distance like this, but hopefully when I will come to Indonesia in
yeah,
Brawijaya, we will be able to make some practical course where the
yeah,
student could handle some modeling part and use some coding for instance, we have here specific courses for students to make them understand well how we use crop model to answer specific questions. So hopefully one day we will be able to do the course in Indonesia as well.
yeah. Of course, maybe this year, right? And Rafael, another students, yeah, they were waiting you at the times, but the student in front of you today online were not student at the time that that hope you come for the first time last year, I think. you know
Okay.
so this is different students that
Yeah.
we were expected for your course at the times, because the course has been already closed for them, and then now we are coming for the new semester, and so that's why this is the new group that before they were not study previously concerning your methods.
Okay. Yeah. So it's even harder to understand.
Yeah. Okay, I don't know if you want to say something as concluding remark, Rafael. As
No,
no,
no. Thanks for the invitation, and I hope we will
okay,
be able to make other course and maybe some basics course on physiology. I had no time to prepare something specific to explain you better the basis of physiology and modeling. And so it was just a view of what we are doing here in Montpellier. But probably next year we'll do a course that is maybe more interesting for the student to better
yeah,
harness or handle
perfect.
the ecophysiology aspect of research.
Okay,
Okay.
very good. Yeah,
Thank
very
you.
good.
Terima kasih.
Thanks, Rafael. So, Hannah, you can close this meeting. Thanks, Rafael.
Thank you.
Thank you, Dr. Perez, for sharing your valuable knowledge and insight with us today. Your expertise in combining modeling and land mapping has provided us a deeper understanding of assessing the feasibility of developing crop environmental interaction. Was an enlightenment also for us. We would also like to express our gratitude to the audience for their active participation and engaging questions. If you have any further queries or would like to connect with Dr. Perez, please feel free to reach out to us. And this concludes our meeting. And we hope you found it informative and inspiring. Thank you all for attending and have a wonderful day. Wassalamualaikum warahmatullahi wabarakatuh.